Good morning, everybody. Tuesday was World Asthma Day. Asthma is the most common chronic respiratory disease in children worldwide. And certainly it's a problem in Pittsburgh, where 22% of kids attending public schools have asthma. That's why, if you ever wonder, that's why you see so many kids affected with this disease. And which better way to celebrate World Asthma Day than to have Dr. Jason Debley with us? Dr. Debley graduated from Northwestern Medical School, completed his residency in pediatrics at Children's Memorial at Northwestern University, went on to serve chief residency at Legacy Emmanuel Children's Hospital in Portland, and then completed his fellowship in pediatric pulmonology at Seattle Children's and the University of Washington. He's stayed there for now over 2 decades and rose to the rank of professor. He also is the director of the Center for Respiratory Biology and Therapeutics of Children's Research Institute in Seattle. Dr. Debley has authored or co-authored 74 original research reports. It's PI of an R1 and MPI of a U19 from the National Institutes of Health. It's truly a pleasure and privilege to have Dr. Debley here. Jason?
So thank you, Dr. Celadon, for that very kind introduction and for the invitation to speak here today. It's really an honor. So I unfortunately have no relevant financial disclosures. So as Dr. Celadon already mentioned, this audience is quite familiar with asthma, and you know that it's the most common chronic condition of childhood, affecting about 1 in 12 children in the United States, or about 6 million kids, and it creates significant morbidity, including over 700,000 ED visits, more than 300,000 hospitalizations, and 14 million missed school days, with a huge cost to the economy here in the United States. And of course, worldwide, those numbers are magnified significantly. And despite the significant improvements in asthma treatments over the past couple of decades that clearly reduce symptoms and exacerbation risk, there's still millions of viral-triggered exacerbations that occur annually, and there's no cure, of course. So to really understand where the field is today, it's helpful to briefly look back. In the mid to late 90s, when I was a pediatric resident, and through the early 2000s, the airway epithelium in asthma was really largely viewed as a passive barrier tissue. And the dominant model of pathophysiology of asthma at the time was really more focused on adaptive immune dysfunction, particularly T2-driven inflammation, which is clearly important. But the structural changes that we see in the airways, like goblet cell hyperplasia, basement membrane thickening, and smooth muscle hypertrophy, were really at the time considered more end organ injury. However, over the past couple of decades, our understanding of the airway epithelium has really fundamentally changed. Rather than just a passive barrier, we now recognize that it's really a dynamic and active participant in pathophysiology, with dysregulated epithelial repair and dysregulated wound healing that at the time was in the early 2000s hypothesized to be more central to asthma, but that was really a conceptual breakthrough. And these are a couple of figures that I pulled from 2007 from Stephen Holgate, who was really a thought leader at the time in advancing our thought about the epithelium. And so now, if you fast forward a couple of decades, we know that the epithelium is really this coordinator of immune responses that senses environmental triggers and then releases signals such as TSLP that influence downstream immune pathways, including T2 inflammation. But this shift from barrier to dysregulated repair to immune regulator really opened the door to understanding asthma as a disease that's really rooted in epithelial biology, not just immune dysfunction. So around the same time, genome-wide association studies began to identify genetic loci associated with asthma. And this slide is from a Sentinel meta-analysis of GWAS studies published back in 2011 that showed that many of the strongest replicated signals from those GWAS studies that I have here boxed in red map to genes like IL-1, RL1, TSLP, IL-33, which were all really tightly linked to epithelial innate immune pathways. So that at the genetic level, we started to see that asthma susceptibility was really closely tied to epithelial-derived signaling and not just immune regulation. So trailblazers like Stephen Holgate, studying the role of the epithelium in asthma around that time, had a really big influence on my early faculty research interests. And together with some distant mentorship and encouragement from folks like Steve Stick in Australia and Daryl Knight, who was in British Columbia at the time, but also Australian. With their encouragement, I launched a pediatric airway epithelial research program at Seattle Children's back in 2007 that really has steadily grown over the past couple of decades. And what we do is we recruit kids who are undergoing elective surgeries that require an endotracheal tube, and we do bronchial brushings. And then we expand and bank those cells. And then we differentiate them at an air-liquid interface, or ALI, into a beautiful organotypic cultures that closely model the in vivo airway epithelium. And then what's unique, a lot of people now use this technique. At the time, it was quite a breakthrough. But what we do that's unique is we deeply phenotype all of these kids. So that includes lung function measurements, exhaled nitric oxide or FeNO. We do allergen sensitization testing, eosinophil counts, and we follow these kids longitudinally to track incident exacerbations and changes in lung function over time. And that really allows us to directly link mechanistic experiments that we do in these models to clinical outcomes. So that's what's really unique. To date, we've enrolled hundreds of kids with and without asthma into this pipeline. That's really now a unique platform to study lower airway epithelial biology in asthma. So using this model system, some of our early studies really focused on how airway epithelial cultures from children with asthma differ from a baseline from healthy kids. And on the left in this study, we found that epithelium from asthmatic kids demonstrates increased expression of a couple of key pro-remodeling cytokines. And that suggested that aspects of airway remodeling may be intrinsic to the epithelium itself. And these are a couple of passages outside of the host. So that was early on suggesting memory. So then we started to ask how these cells respond to viral infection. In the study on the right, you can see that respiratory viral infection, in this case RSV, induced epithelial production of TSLP, an important upstream alarmin regulator of T2 inflammation. And that response is amplified in the kids with asthma and further amplified in the context of viral infection, in this case RSV. And that was that study was the first to show in pediatric epithelium that TSLP was differentially expressed in asthma and augmented by viral infection. So now if we fast forward to 2026, we recognize that the airway epithelium is really, as I like to say, the center of the universe, but more a central regulator of asthma pathophysiology. And it really does sit at this interface with the environment. It senses asthma triggers like allergens, viruses, bacteria, and as well as air pollution. And then it responds by releasing these upstream immune signals, or like we'd like to say alarmins. And TSLP, IL-33, and IL-25 are sort of the key alarmins. And then these epithelial signals then coordinate the downstream immune responses, including T2 inflammation, eosinophils, and other immune cells, IgE production. And of course, also that leads to the structural changes that we see in asthma. So today, many of the biologic therapies for severe asthma that we use, including mepolizumab, dupilumab, and tezepelumab, all target pathways that really originate from epithelial signaling. So what have airway omics studies to date taught us about asthma? So there's a lot that I could potentially talk about today, but I'm just going to touch on a couple of really key sentinel papers. The shift towards viewing epithelial biology as central to asthma really has been advanced by some key airway transcriptomics studies. And in this landmark study by Woodruff and colleagues at UCSF, genome-wide profiling of the airway epithelial cells, these were all adults, identified a set of genes that are highlighted here in red, including CLICA1, periostin, and serpin-B2 that were highly expressed in a subset of patients with asthma. And importantly, this signature now we understand reflects IL-13 driven T2 inflammation, and it's associated with clinical features like eosinophilia and responsiveness to corticosteroids. So this really was a landmark study that for the first time showed that asthma is not a single disease, but really consists of distinct molecular endotypes. And you can see from the figures from that same paper where they studied the cells in vitro that IL-13 strongly induces expression of these three genes, periostin, serpin-B2, and CLICA1, which really are the key components of this signature that's now really recognized as a canonical molecular T2 high endotype in asthma. And importantly, this signal is suppressed by corticosteroids, which helps explain why T2 high asthma tends to respond more to steroids. And this IL-13 epithelial program has really, as I mentioned, become foundational to how we define and treat asthma, but it's not the whole story. Subsequent transcriptomic studies really have expanded this concept beyond thinking of asthma as just being divided into T2 high versus T2 low. This is a key study from the U-BioPred cohort, which is a large multi-center adult severe asthma cohort. And in this study, they identified 4 distinct groups with different underlying biology and clinical characteristics. 2 of the groups from their study in this table, group 1 and group 3, really represent distinct eosinophilic endotypes. Group 1 here showed high submucosal eosinophils on bronchial biopsies and high FeNO, exhaled nitric oxide, and frequent exacerbations and high steroid use. Whereas in comparison, group 3 showed the highest sputum eosinophils and a different clinical profile, including higher BMI. So even within what we might all clinically lump together as T2 high clinical asthma, there are different biological programs driving the disease. And then they showed in this group labeled 4 here that nearly half of their patients really had little evidence of eosinophilic or immune activation at all, highlighting how heterogeneous this disease that we all lump together as asthma really is. So here from the same study, the subjects are grouped based on pathway level gene expression. And to orient you to this heat map, the group of patients on the right side with broad red signal across multiple pathways that you can't see, it's not labeled here, but trust me, that's T2 inflammation, Th2, interferon, and steroid-related pathways. And that group corresponds to patients with more severe eosinophilic disease, but you can see a lot of heterogeneity. And then in contrast, patients on the left in green reflect lower overall pathway activation. So really this is kind of a confusing study when you dive into it, but one of the key takeaways here is that asthma endotypes could be defined by coordinated activation of multiple different immune pathways rather than a single pathway. These kind of transcriptomic approaches have also been extended to kids using less invasive sampling strategies. And this is a sentinel study from Max Siebold's group and others that really they profiled kids with asthma, and they were able to also distinguish T2 high and T2 low molecular profiles, similar to what had been shown in adult bronchial studies. But what's important here is that, again, this separation is being driven by the underlying epithelial and mucosal biology, not by clinical features, again, reinforcing this concept of molecular endotypes. And critically, this was performed with nasal samples. So this was a really landmark study to show that you could use nasal sampling as a proxy for the lower airways, and that you could apply it into larger cohorts longitudinally and in real-world settings like during exacerbations. So building on transcriptomic approaches, epigenomic profiling has also further advanced our understanding of airway biology in asthma. And this is a particularly important large study by Eric Farno and Juan Celedon's leadership here, of course, in Pittsburgh. And DNA methylation in the nasal epithelium was examined across several large pediatric cohorts. And they identified distinct methylation patterns that were associated with atopy and atopic asthma. And again, demonstrating that the airway epithelial biology really reflects underlying disease endotypes. And importantly, they replicated these findings across multiple populations, highlighting the robustness of these signatures. And you can see this in this slide on the left. They found widespread differences in methylation across the genome that associated with atopy. And in the middle, these differences really cluster into distinct patterns of hyper and hypomethylation. In the right, they have an ROC curve here that showed that these methylation signatures can actually be used to predict atopy with fairly high accuracy, suggesting potential utility as biomarkers. So if you put this all together, this study really showed that nasal epithelial epigenomic profiling is not only biologically informative, but also could have clinical utility for risk stratification. So more recently, transcriptomic profiling in nasal samples has continued to refine our understanding of asthma endotypes in kids. And this is another study published by Juan Celadon's group and colleagues last year in JAMA. And the gene expression profiling across multiple cohorts, again, pediatric cohorts, identified 3 really reproducible molecular endotypes, a T2 high group, a T17 high group, and a T2 low, T17 low group. So what's really striking is that these patterns are consistent across these independent populations, reinforcing the robustness of these signatures. But perhaps most surprising is that T2 high was not the dominant endotype of these cohorts, which really challenged kind of current dogma about kids with asthma. And this suggests a substantial portion of asthma may have non-T2 driven disease, which is, of course, less well understood and less well targeted by our current therapies. So how did current clinical biomarkers perform in identifying these molecular endotypes in their study? Here you can see the performance of things that we commonly use in the clinic, blood eosinophils, FeNO, and total IgE to predict T2 high biology. And while these markers were okay in the study, their accuracy was pretty modest overall. And importantly, they lacked sensitivity. So a significant number of patients with T2 high biology in the airway may not be identified. And this highlights a really key limitation of our current clinical approach. We're using relatively crude clinical markers to approximate way more complex biology. So given that up to 90% of exacerbations in kids with asthma are triggered by viral infection, and the vast majority of those are rhinovirus, as you all know, what have airway omics studies taught us about viral-triggered exacerbations? So to move beyond static endotypes, this is a key transcriptomic study by Matt Altman and colleagues where they asked whether airway biology changes during viral infection that leads to asthma exacerbations in kids. So they used longitudinal nasal profiling during naturally occurring colds to identify distinct molecular networks associated with exacerbations. And this is a pretty complex paper. I'm not going to go through all the detailed findings and gene modules, but at a high level, some of the differentially expressed gene modules reflected expected pathways, such as type 2 inflammation and epithelial remodeling. But they also identified exacerbation-specific modules, including type 1 interferon responses and innate immune pathways that were differentially expressed between the kids who had asthma exacerbations triggered with viral infection versus not. So this study really was one of the first to define molecular programs associated with exacerbation risk in kids with asthma, not just baseline disease. So what was particularly powerful about this study is that it showed how these programs evolve during time, during a viral infection. And on the left, you can see that kids who go on to exacerbate show really distinct trajectories in pathways such as epithelial remodeling and inflammatory signaling compared to the kids that did not. And in the middle, in purple there, virus-associated responses, particularly type 1 interferon signaling, also differed between the groups. And on the right, the molecular signatures were associated with differences in exacerbation risk over time. So this really raised some important questions. Are these differences in antiviral responses intrinsic to the airway epithelium? And can they help explain why some kids are really exacerbation prone versus those that are not? So this is a more recent study from Nero Hershey's group in Cincinnati and collaborators that extends the concept by asking whether airway transcriptomics from nasal brushings can really identify kids who are prone to frequent exacerbations. And here they used nasal samples and RNA-seq, and they compared frequent exacerbators to non-frequent exacerbators, and then identified distinct transcriptional networks in each of these groups. And what they found was that children with frequent exacerbations showed enrichment of pathways related to type 2 inflammation, as well as epithelial defense responses, while non-frequent exacerbators showed signatures more consistent with epithelial differentiation and ciliary function. But there's a really important limitation, from my vantage at least, to keep in mind in this study. Samples were obtained during or shortly after a clinical exacerbation. Often after even treatment had been started. So while they could identify associations with exacerbation-prone status, they really couldn't determine whether those patterns reflected any predisposition to exacerbation, or simply it was reflecting the sort of aftermath or tire tracks of the treatment effects. So this really illustrates, I think, an important concept. Without baseline sampling and without capturing the responses to viral infection over time, you also can't define the underlying antiviral or epithelial state that might explain that exacerbation risk. So it highlights a really key gap in the field. We need to move from static or post-event snapshots that might be easy to do in a study, but we need to understand the pre-infection biology and then the dynamic responses to viral challenge. So to begin to address these questions, we and other groups have been focused on the airway epithelium as really the central regulator of antiviral responses. So as we all know, when respiratory viruses infect the airway, the epithelium really acts as that first defense. And there are all kinds of viral sensors that I won't go into the details today, but they end up sensing that infection and then producing interferons that drive antiviral programs, including hundreds, if not thousands, of interferon-stimulated genes or ISGs. And these early responses ideally help contain the viral infection at the mucosal surface, but they're also intended to sort of signal the underlying immune system to amplify the response if it's needed. So a key question, and that one that has been endlessly debated in the field for over 20 years, is whether these epithelial interferon responses in asthma are deficient, are they delayed, are they dysregulated? So some of the earliest studies in this area suggested that the airway epithelial interferon response might be sort of generally deficient in asthma. And in these two landmark papers, bronchial epithelial cells from from adults with asthma were infected with rhinovirus in vitro. And I will say that the model systems they used were not differentiated. They were much more primitive model systems at the time. And they compared responses to healthy controls and showed what seemed to be a reduced production of interferons. This led really to this hypothesis that impaired epithelial antiviral responses could result in increased viral replication and then greater susceptibility to exacerbations. But really, that model turned out to be really incomplete. So subsequent studies from multiple groups reported that airway epithelial cells from individuals with asthma can really mount a normal interferon response, particularly in well-controlled disease. And this suggested that maybe the antiviral response in asthma was not uniformly deficient, but maybe was more heterogeneous and context dependent. So this question was also tested clinically in a randomized controlled trial of inhaled interferon beta for viral-induced asthma symptoms. And while there was some signal in secondary outcomes in subgroups, overall, this trial showed pretty limited efficacy. But there were some critical limitations in that trial that we need to remember. The study was relatively small. Subjects were not specifically selected for highest risk for exacerbation. And probably most importantly, treatment was initiated days after symptom onset, which is likely far too late to meaningfully modify antiviral response. So taken together, the question has really evolved over time from whether interferon responses are simply deficient to really understanding how they're regulated and when they occur and in which patients they matter most in. So to address this question more directly, we've been studying antiviral responses in our primary bronchial epithelial cultures from kids with and without asthma for some time now. And this is a study from 2018 where we differentiated bronchial epithelial cells into an organotypic state at ALI, and then we infected them with RSV. And then we performed transcriptomics profiling along with protein measurements to really try to characterize that antiviral response. And then within the asthma group, we stratified kids based on lung function, specifically whether they had airway obstruction or normal lung function. And this really, we were trying to ask whether epithelial antiviral response was not just different between asthma and healthy, but whether it varied within asthma based on disease severity. So when we looked at epithelial response to RSV, we saw not a deficient interferon response, but actually a heightened response in a subset of children with asthma. So starting on the left, this volcano plot shows differential expression comparing asthmatic kids with airway obstruction to those without. And what stands out is a really strong enrichment of interferon-stimulated genes, or ISGs, indicating a pretty robust antiviral response in the obstructed group differentially. And on the right, we summarize the interferon at a pathway level. So this is interferon plus a whole bunch of ISGs. And that response was actually highest in the asthmatic kids with airway obstruction, there in red. And we use 2 different strains of RSV. And at the bottom right of the slide, you can see a strong inverse relationship between interferon response and lung function. So a higher interferon signaling was associated with lower FEV1. So these data all together really suggest that in this context, at least, post-infection antiviral response by the epithelium was not deficient, but was exaggerated and tracked with disease severity. So now I want to give a little bit of a setup for work we've been doing for really the last 8 years or so, trying to characterize bronchial epithelial innate immune responses to rhinovirus in childhood asthma. So as I described before, we collect bronchial brushings from pediatric patients with asthma and healthy children, and we differentiate these at an ALI to develop organotypic pseudostratified cultures. And then we assess their baseline transcriptome and protein production, and then we infect them with human rhinovirus. And in our case, we use rhinovirus A16. And then we assess over sort of a rich kinetic time course responses by both bulk and single-cell-seq, as well as protein production for up to 10 days after infection. So for this first story I'm showing you here, which we published in JCI Insight late last year, we used bronchial epithelial cells from pediatric patients with asthma, those with both a history of severe exacerbations and not, as well as some healthy controls. And importantly, these 2 asthma groups were well matched across a variety of clinical characteristics like age and sex and lung function and treatment exposure. And notably, there were really no differences in traditional type 2 inflammatory markers between severe exacerbators and non-severe exacerbators. So the first thing we asked here was whether viral replication differed across these groups. And on the left, you can see the viral load over 10 days following rhinovirus infection. And what stands out is that the epithelial cultures from the kids with a history of severe exacerbation there in red show consistently higher viral loads compared to the non-severe asthma and healthy groups. And this group really, this difference really persists over this whole 10-day time course. So it's not just a delay or a timing issue. These cells really appear to be intrinsically more permissive to viral replication. And to orient you here, this is on a log scale. So these are actually significant differences. So taken together, this really suggests that kids with severe exacerbation history have an epithelial endotype that supports greater viral replication. So to better understand what's driving these differences in viral replication, we looked at RNA-seq across this time course from baseline through 10 days after infection. And then we used a modular analysis approach to identify groups of genes that change together over time and compared those patterns between the kids with and without a history of severe exacerbation. And what you can see is that we identified a set of modules that differed significantly. And from those, we really focused on 5 modules that were of greatest biologic interest to us, including interferon response, stress response, epithelial remodeling, and then cellular metabolism. So here I'm highlighting just 2 of those modules, the interferon response and cellular stress response. And on the left of each of these 2 modules, you can see their expression over time following infection and GAM plots. And what stands out is that the kids with a history of severe exacerbation in red showed a greater and more sustained upregulation of both these pathways compared to the kids without severe exacerbations and the healthy controls. And on the right for each of those 2 modules, you can see that the magnitude of those responses really strongly correlates with the viral load. So rather than a deficient response, what we're seeing is really an exaggerated interferon response and persistent inflammatory response that tracks with the degree of viral replication. So together, this really suggests that in these kids, the airway epithelium is mounting a heightened response, but one that might be ineffective in controlling the virus and instead maybe contributing to disease severity. So in contrast to the increased inflammatory responses, we saw metabolic and cellular transcriptional pathways that were suppressed in the kids with severe exacerbations. And that suppression was deeper and more sustained in the severe exacerbation group, and in this case, inversely correlated with viral load. So then we asked whether baseline differences in the airway epithelium might explain the differences we see after infection. So specifically, we looked at what we call pre-infection interferon tone. So on the left, you see gene set enrichment that shows that interferon response pathway is lower at baseline in the kids with a severe exacerbation. And to translate that into a measurable protein signal, because the interferon beta and lambda are quite difficult to measure, we focused on CXCL10, a well-established and easy to measure across a dynamic range, ISG. And in the middle panel, you can see that the pre-infection CXCL10 levels are substantially lower in the severe exacerbation group compared to both non-exacerbators and healthy control. And importantly, on the right, those baseline CXCL10 levels were inversely correlated with viral load after infection. So if you put this all together, it suggests that baseline interferon state predisposes the epithelium to this greater viral replication, which then leads to an exaggerated downstream interferon and inflammatory response following infection. So really, in other words, rather than a failure to respond to infection, this appears to be more failure to prepare for infection in these kids. So this led us to ask really a key mechanistic question. Is low baseline interferon tone actually driving this increased viral replication we're seeing? So to test this, what we did is we treated these epithelial cultures prophylactically with interferon beta prior to infection to really try to restore interferon tone and then infect it with rhinovirus. And here, what you can see is that prophylactic interferon beta markedly reduced viral replication. And importantly, we see this in both groups, but it really normalized the higher viral load in the severe exacerbation donors. So this provides some functional evidence that this baseline pre-infection interferon deficiency is not just associated with, but likely more causal for the increased viral replication. So then we asked whether restoring interferon tone not only reduces viral replication, but how did it impact the downstream epithelial responses we observed? And this is a busy slide, I realize, but across the same pathways that we looked at, I showed you before, interferon signaling, inflammation or remodeling, stress responses, and metabolism, the interferon beta prophylaxis really shifted that response towards a more normalized state. Next, we looked at responses at single cell level across epithelial cell types. And I don't have time to show you all of the different modules here, but I'm just going to show a couple. Red indicates higher expression in severe exacerbation donors. And we see that interferon response genes are pretty broadly increased in the severe exacerbation donors across cell types, but are a little stronger in specific populations: basal cells, secretory cells, ionocytes, tuft, and ciliated cells. In a little bit different pattern, in contrast to interferon response, the stress response is a little less broadly distributed, but strongest in basal cells, ciliated cells, and tuft cells. So to put this in clinical context, I want to highlight a recent prospective study from the Intercity Asthma Consortium, Muppets 1 cohort, also published last year. First author, Courtney Gabarino. In this study, children with exacerbation-prone asthma were followed over time with nasal samples collected at baseline and then during naturally occurring viral illnesses. And what they found was a strikingly similar pattern to what I showed you in our pure bronchial epithelial model. Children who went on to severe exacerbation had lower baseline interferon expression, but then mounted an exaggerated interferon response early during infection. And that heightened response was associated with higher viral load, worse lung function, and more severe symptoms. And importantly, lower baseline interferon tone also predicted a shorter time to the next exacerbation. So if you take this all together, these in vivo nasal data are pretty closely aligned to what we see in our in vitro pure bronchial model. That low pre-infection baseline interferon tone really predisposes to viral replication and then is followed by an exaggerated, dysregulated response after that probably is pretty key to driving the exacerbations. So together, these studies really support a low baseline interferon tone exacerbation-prone endotype that really cuts across T2 high and non-T2 clinical phenotypes. which is pretty interesting. And then taking from our study, given that prophylactic recombinant interferon reduced viral load and normalized these post-infection responses, it really begs the question, is there a role for prophylactic recombinant interferon targeting patients with this low baseline interferon tone endotype, particularly given the limitations that I told you about the trial from a dozen years ago? So briefly, I'd like to highlight an adjacent study looking at how IL-13-driven type 2 inflammation alters airway epithelium prior to viral infection. In this study, we used bronchial epithelial cells from 61 pediatric donors, and we differentiated them at ALI, I described before, and then conditioned them, in this case, with IL-13 for 7 days before rhinovirus infection. And what we found here is that IL-13 induces substantial epithelial remodeling. It induces significant expansion of goblet mucus-producing cells and also causes some disruption in ciliary structure. Now, if Sally Wenzel from Pittsburgh was here, she'd say, yeah, of course, that tracks. We also looked at how IL-13 alters transcriptional responses before and after rhinovirus infection. And on the left, you can see that IL-13 alone induces pretty widespread transcriptional changes in the epithelium, reflected in the volcano plot and in the yellow column of the heat map below it. But if you focus in, there are three key effects that emerge. First, the interferon response and the regulatory pathways are suppressed in the presence of IL-13. And then second, the antiviral effector programs seem to be blunted, suggesting a reduced capacity to control the viral infection once it's started. And then third, we see an enhancement of secondary pro-inflammatory pathways. So overall, the IL-13 shifts this epithelial response away from an effective antiviral defense and toward more inflammation. And that provides a nice mechanistic link to what we've been discussing, where T2 inflammation might lower baseline interferon tone and predispose to the exaggerated response following infection. So this is an in-progress study. We've been looking at gene expression at the single cell level in response to IL-13. And on the left is a UMAP that shows 11 distinct epithelial cell populations in these bronchial epithelial cultures. And on the right, we can quantify the cell populations. And IL-13 really shifts this epithelial landscape. Most notably, there's a marked increase in goblet cells, and along with reductions in club and some other progenitor populations. So IL-13 really drives this mucus-skewed epithelial phenotype even before infection. So we next asked how IL-13 affects antiviral signaling at the signal cell level. And the goblet cells is where a lot of the action is. And of course, that's the cell population that I just showed you was markedly expanded in a T2 IL-13 environment. And what we see here is reduced expression of type 1 and type 3 interferons, as well as some key viral sensing receptors, toll-like receptors. And then on the right, more broadly across all the epithelial cell types, we see that IL-13 downregulates interferon receptor expression. So IL-13 is really impairing both interferon production and the ability of the cells to respond to interferon. And then finally, at the bottom here, when we look at viral load in the single cell experiment, we see increased viral load with the IL-13 conditioning. So taken together, IL-13 not only reshapes epithelium with increased mucus-producing goblet cells, but also seems to create an impaired antiviral state across epithelial cell types. So really, I promise this is the last data slide I'm going to show you. This is data from a recently published brief report highlighting some of our ongoing work, really trying to examine how IL-13 and rhinovirus, each in their own way, may drive persistent changes in the epithelium through epigenetic regulation. And this is really trying to pair DNA methylation with transcriptomics data. And here, both IL-13 and rhinovirus infection induce widespread changes in DNA methylation across bronchial epithelial cultures at loci that are relevant to asthma biology. And here I'm showing a couple of genes, SERPIN-B2 and RHO-B, where these methylation changes are associated with altered gene expression, suggesting that functional consequences of that DNA methylation. So these exposures may also reprogram the epithelium at an epigenetic level. And as we know, this could be happening over many years in a child's life. So this raises that possibility that both T2 inflammation and viral infections could leave really this durable imprint that affects epithelial function over life. So what are our next steps? Well, together with collaborators, Matt Altman at the Benaroya Research Institute in Seattle, and who's also very embedded in the NIAID Intercity Asthma Consortium, and Carol Ober at the University of Chicago, we're really working to leverage this carefully phenotyped and genotyped and longitudinally tracked pediatric asthma cohort, where I didn't show you any nasal data, but in every single kid that we recruit, we have paired nasal and bronchial fresh brushings. And then, of course, we can take those all to ALI with the goal over the coming years to really tackle several aims in the context of rhinovirus-triggered asthma. There are many more than this, but these are three that I'm highlighting. We really want to define ex vivo, so those fresh brushings, airway epithelial interferon endotypes and their genetic and epigenetic determinants across both nasal and bronchial sites. Because obviously we use nasal as a proxy, but there are things that are clearly different. If you think about it, nitric oxide is much, much higher or constitutive nitric oxide between the upper and lower airway. And there are many other things that are different, even though clearly there are things that are preserved. And then we want to look at T2 cytokine-driven plasticity of these interferon endotypes in these paired nasal and bronchial ALI models. And then really, this is what's most interesting over time, is what would be the impact of recombinant interferon prophylaxis on these interferon endotypes. So hopefully, to try to tie this all together, because I've gone through a lot here, hopefully I've convinced you that airway omics studies have really significantly advanced our understanding of asthma pathophysiology over the last couple of decades. They've identified novel therapeutic targets. They've identified specific molecular endotypes, and obviously that's still evolving. They've identified biomarkers within this heterogeneous disease that we all sort of lump together as asthma. And really all together, I'm not going to go through this entire table here, but they're moving us much closer to this ultimate goal of precision medicine, where in the future, and probably not too far from now, we will, instead of relying on peripheral eosinophilia on blood samples or pheno alone, we will do nasal brushings and have a really comprehensive multi-omic approach to decide what is the right therapy for each kid. So that's where I'm going to finish. I do want to thank my lab, which I'm very blessed to have a very loyal and talented group of staff scientists and physician scientists that help us with all this work, and research coordinators. And I want to call out the Altman Lab, Genomics and Bioinformatics Core, led by Matt Altman there in Seattle. And of course, our funding from NIAID. And really, of course, want to thank all of the children and families who participate in our studies, because none of this would be possible without them. So I'll finish there. Thank you very much.